Hi everyone, good afternoon. My name is Joseph Okelo Rael. I'm a student at Ines Ruhenjeri doing biomedical laboratory sciences. My rec number is 23 20995. So I'm here to present question number 31, which reads, what is the principle behind agglutination reactions? So agglutination reaction rely on the principle of cross-linking. Antibodies or other agglutinants bind to multiply antigens, typically on the surface of the cells. Um, Antibodies, this type of cross-linking leads to the formation of visible clones or aggregates. So therefore, cross-linking of antigens by antibodies resulting in visible grouping. So in this question, like the principle behind agglutination, like when I talk about agglutination, it is the reaction between antigen and antibody in the immune system of a living organism. So my second question is question number 32. So it says, how does lattice agglutination work in the diagnostic? So lattice bits are coated with antibodies specific to a target antigen. The sample containing the potential antigen is mixed with the antibody coated uh, leads to bits. So if the antigen is present, it binds to the antibodies on the bits, causing agglutination. So agglutination is a positive result indicating the presence of the targeted antigen, or absence of agglutination is a negative result. So like once you did your reaction, so the presence of positive results, there will be agglutination. So if it is a negative result, agglutination will not occur. So this is the meaning of the latex agglutination and how it works in the diagnostic of antigens and antibodies. So I'm going to continue with the third question. So the third question is, what is the principle of precipitation reaction in immunology? A precipitation reaction involves the interaction between soluble antigens and their corresponding antibodies. The formation of antigen-antibody complex occurs when the ratio of antigen to the antibody is optimal. These complexes become insoluble and precipitate out of the solution, forming a visible precipitate. So therefore, the formation of a visible precipitate is due to the interaction of the soluble antigens and the specific antibodies at the optimal options. So to continue with my 34th question, so it is like, how is the radial immunodiffusion test performed? So a gel containing antibody is prepared. Wells are put into the gel, then the samples containing the antigens are placed into the wells. Then after that, the antigens will diffuse radiantly outwards from the well. Then a precipitation ring forms where the antigens-antibody ratio is optimal. The diameter of the precipitation ring is proportional to the concentration of the antigen. So what does it mean here? So it means that the antigen containing the samples are placed in the wells in an antibody containing gel. Then the radial diffusion of the antigen leads to the precipitation ring formation, with the ring diameter indicating antigen concentration. So my last question is like under the principle of ELISA technique. Like when I talk of ELISA technique, ELISA stands for enzyme linked immunosorbate assay. Like it uses an enzyme linked antibody to detect the presence of an antigen or antibody in a sample. The enzyme catalyzes a calorimetric reaction, allowing for qualitative and quantitative detection. The intensity of the color is proportional to the amount of antigen or antibody presence. What I mean here is like, uh, it's like an enzyme linked antibody detects the presence of what, of the antigen or antibody. So the catalyst will form the reaction, then the intensity of which it is proportional to the target concentration. So in conclusion, I'm going to conclude as I have presented about agglutination. It is generally like the reaction between antigens and antibodies in the immune system of a human being. So thanks, this is my presentation.